Let's go to the next presentation. This will be from Dr. Elizabeth Demicco with a title of Intimal Sarcomas as it pertains to molecular landscape and profiling. So Dr. Demicco is our bone and soft tissue pathologist and section head of surgical pathology at Mount Sinai Hospital in Toronto, Toronto. She is also an associate professor at the Department of Pathology at the University of Toronto. She earned her MD and PhD both at Boston University School of Medicine and MGH respectively. And she was a fellow in MD Anderson in Texas. Before taking her appointment at Mount Sinai Hospital in Toronto, she was in Mount Sinai Hospital in New York, where she was also the director of the bone and soft tissue section and the head and neck services up to the year 2017. Without further ado, I would like to invite Dr. Demicco to present her talk.
Hello, my name is Elizabeth Demicco. I'm a staff pathologist at Mount Sinai Hospital and associate professor at the University of Toronto. I'd like to thank the organizers for inviting me to speak today on the molecular landscape of intimal sarcoma. I'm going to give you a brief background on intimal sarcoma, talking about the definitions of intimal sarcoma and cardiac sarcoma, a little bit about the anatomy to discuss why there's some controversy on what constitutes an intimal sarcoma. Then we're going to talk about the molecular features both of vascular intimal sarcoma and quote-unquote cardiac undifferentiated sarcomas before bringing you to our conclusions. First thing that you have to understand about intimal sarcoma is that there's some controversy as to what actually comprises an intimal sarcoma. As you can see from these definitions taken from the current edition of the WHO, some people consider intimal sarcomas to only represent tumors that arise within arteries, whereas tumors that arise in the heart they would consider to be more properly called cardiac undifferentiated pleomorphic sarcomas. However, soft tissue pathologists use the term intimal sarcomas for both because we're very flexible about our definitions and often are used to names that don't necessarily apply, strictly speaking, to anatomic sites, which we'll discuss in the next slide. The question then becomes, are cardiac sarcoma and intimal sarcoma even the same thing? And the debate comes down to the anatomic structures lining the blood vessels and the heart. So the vascular intima obviously is comprised of endothelial layer overlying a layer of actin expression quiescent parasitic-like cells, which are considered to be pluripotent mesenchymal precursors. Underlying this is a vascular smooth muscle parasitic layer with more myogenic differentiation. There's also, of course, an admixture of resident immune cells within the intimal layer. The cardiac endocardium, on the other hand, is comprised of a variable mixture of endocardium, which is fibroblasts and smooth muscle, and an admixed subendocardial layer, which may be mixed with the endocardium or underlying it, which includes Purkinje fibers, nerves, blood vessels, undifferentiated mesenchymal cells, fibroblasts, and macrophages. However, if you look at the tumors that arise from intima, we see angiosarcoma arising from endothelial cells. We have undifferentiated intimal sarcoma, which is variously called in the literature myxofibrosarcoma, UPS, or MFH. And there's intimal sarcomas that have some line of differentiation, often described as being rhabdomyosarcoma or osteosarcoma, etc. And it's unclear if these are really distinct entities or if it's just a sarcoma that has heterologous elements, analogous to like a dedifferentiated liposarcoma with heterologous differentiation type of. Very similarly, the sarcomas arising from the cardiac endocardium include angiosarcoma, undifferentiated sarcoma, and sarcomas with some line of differentiation, most typically including rhabdomyosarcoma or osteosarcoma. And again, unclear if these are heterologous elements or not, or if they're distinct entities because there hasn't been a lot of work done on characterizing these. Histologically, intimal sarcomas are characterized by their intraluminal growth and protrusion into the vascular lumina. They have a variety of morphologic features. In this example illustrated here, we see a fascicular tumor comprised of bright eosinophilic spindle cells, which show very much smooth muscle, sort of myogenic differentiation. Tumors may show more haphazard architecture, frequently with pleomorphic cells dispersed within an abundant myxoid background, either fascicular architecture or just kind of sheet-like growth. And in fact, some tumors may show perivascular accentuation with more dense tumor cellularity around areas of small intratumoral blood vessels, as seen here in the lower right, panel E. While tumors do protrude into lumens, they also can invade into the wall of vessels and from there into surrounding soft tissue, as illustrated in panel B in this figure taken from Bo Lesniewska et al. In 2001. Very similarly, cardiac undifferentiated sarcomas, or intimal sarcomas as some call them, typically show pleomorphic cells within a mixed. They can be very cellular with eosinophilic cells arranged in fascicles. They can be more pleomorphic, they can be more epithelioid, and they often have very frequent myxoid stroma with various surface. Again, a wide range of appearances in cardiac intimal sarcoma. So let's talk about molecular profiling of arterial intimal sarcoma. I'm going to discuss this base in kind of a timeline format, starting from our very first studies in 2001 using CGH up until the present day using various more targeted molecular profiling modalities. We're going to start off by talking about arterial sarcomas first, and then we're going to talk about studies that either look at cardiac intimal sarcomas or combined cardiac and vascular studies. And I just want to give this timeline to show you the gaps in what we do and don't know and how things have changed and evolved over the past 2 decades as technology has shifted. So let's start with the first series by Bode, Lesniewska et al. In 2001, and also their follow-up study in 2002 using CGH and then array CGH on 8 tumors of their initial series. They found numerous gains in these tumors and a few losses. This is illustrated here in pictorial form. And then in table form, you can see overall there were 71 genetic alterations, including 34 losses, 26 gains, and 11 amplifications. The most frequent gains were seen in chromosome 12q, 1,3 to 1,5, with various losses on 3p, 3q, 4q, 9p, 11q, 13q, xp, and xq, with gains on 7p, 17p, and 17q, and amplifications on 4q, 5p, 6p, and 11q. And then in order to determine in more detail what genes were actually affected, they did a the following year on the tumors that showed gains. So the 6 tumors they performed array CGH on, we see that the most common amplification is actually PGFRA, and this can be very focal, as not all the cases that showed PGFRA amplification on chromosome 4q12 showed amplification in the conventional comparative genomic hybridization study. They also found EGFR amplification in 2 out of 6 cases, and chromosome 12q was amplified in various extents in 6 out of 6 cases, with the minimal common region seeming to be CDK4 in their study, with slightly fewer cases having amplification of MDM2. Subsequently, in 2005, Sebenik et al. Analyzed 17 cases of vascular intimal sarcomas using conventional comparative genomic hybridization. Strangely, they found that only one tumor had any significant copy number changes, and that had numerous gains and losses, mainly involving chromosomes 1, 2, 6, 7, 9, 10, 17, and 20. Strangely, 11 cases had no copy number changes, and that is somewhat conflicting with pretty much every other series out there, so I'm not really sure how to explain that. Another interesting take decided to look at the APC and Wnt pathway in intimal sarcomas, because they were considered a myofibroblastic tumor, and myofibroblastic tumors often have activation of the Wnt pathway. So Gauman looked at beta-catenin mutations and APC mutations in these tumors. They found there was loss of heterozygosity of APC on chromosome 5q to 1.1 in 5 cases, and microsatellite instability in 1 case. There were no mutations in APC and no mutations in beta-catenin. They concluded that APC and beta-catenin pathway changes are rare in intimal sarcoma. In 2019, Wu et al. Performed whole exome sequencing on it. They found that KIT was amplified with 6 copies, as well as p53 mutations on exon 4 and PI3 kinase missense mutation in exon 8 that was presumed to be functional. They did not find any evidence of high tumor mutational burden, mismatch repair, or PD-L1 expression. And this table here illustrates the mutations and copy number variants that they did find. So that was pretty much it for papers that just looked at intimal sarcoma. Moving on to series that investigated cardiac undifferentiated sarcoma. You can see here that most of the activity on cardiac undifferentiated sarcomas has been done in the past 6 or 7 years using array CGH, whole exome sequencing, RNA-seq, methylation profiling, SNP array, and various targeted NGS. But going back to the earlier series, in 2007, Tamburini et al. Looked at tyrosine kinase alterations in intimal and mural heart, including PGFRA, and they looked at the different patterns by FISH of amplification, and said that while some cases have high-level amplification, as seen here at the top panel, other cases have polysomy with more actual centromeric amplification rather than PGFRA amplification. Some patients may have high polysomy, particularly PGFRB, and then EGFR is typically amplified associated with chromosome 7 polysomy. And in many cases, amplification or polysomy is associated with increased protein expression by immunohistochemistry. And they correlated their immunohistochemistry in fish in this table of their 7 patients and saw that in most cases, increased copy numbers of PDGFRA correlated with increased expression by immunohistochemistry. Likewise, increased copy number of PDGFRB correlated with increased expression by immunohistochemistry. EGFR2 polysomic cases typically showed more expression than tumors with low polysomy. However, KIT, despite being amplified or polysomic in many patients, did not correlate with increased immunohistochemical expression of c-KIT. They also looked at activating mutations in these genes and found no activating mutations in PDGFRA, PDGFRB, EGFR, or KIT. They did find that PDGFA and PDGFB, the ligands, were expressed, indicating that there's likely going to be high signaling through these pathways. In 2007, Zhang et al. Performed karyotyping on a single case of intimal sarcoma. Found that multiple chromosomes showed duplication, as well as some marker chromosomes and amplification of, by FISH, chromosome 12q13 to 15, including SAS, CDK4, and MDM2. However, genes that are both telomeric and centromeric of this locus, including ATF1, DDI3, and HMGA, were not amplified. In 2010, Dewal et al. Investigated PDGFRA and EGFR sarcoma. They performed cytogenetic, molecular, and biochemical analysis of 21 patients, including 3 cardiac intimal sarcomas, 15 pulmonary artery, and 3 other vessel intimal sarcomas. The 3 cases they performed karyotype on found the tumors were polyploid with complex structural rearrangements, as well as homogeneously staining regions within unidentified marker chromosomes in 2 cases, and double minutes in the 3rd case. When they performed array CGH, they found the most common losses were in chromosome 3, chromosome 9, and chromosome 10, with chromosome 9 loss of CDKN2A and CDKN2B in 75% of cases. In terms of chromosome gains, 4q12 was seen in 88% of cases involving PDGFRA as the minimal common locus of amplification among these cases. And of course, chromosome 12q13 to 15 was amplified in 75% of cases involving various combinations of focal amplifications in that region. This plot just shows the overall frequency of copy number gains and losses, including chromosome 4, chromosome 6. Chromosome 7 was frequently gained the whole chromosome. Chromosome 8 had a lot of whole chromosome gains as well. Losses were typically seen in chromosome 13 and 18, as well as chromosome 10. They also noted by FISH that there are different patterns of amplification that were seen, as well as amplification or polysomy in different patients, as well as intratumoral heterogeneity, with individual cells within a single patient showing different patterns of amplification. As seen here in panel C, some cells within a cohort showed PDGFRA amplification, while other ones showed MDM2 amplification, and a few cells showed both. Likewise, when they looked at co-amplification of EGFR and PDGFRA, again, some cells showed amplification of one, some cells showed amplification of the other, and some cells showed both, indicating that if you target only one amplified region for one of these growth factors, other cells may continue to proliferate. Overall, they saw that the MDM2 was amplified by FISH in 11 out of 17 cases, and 1 case had polysomy. EGFR, there was gain of chromosome 7p in 6 out of 8 patients, with high-level aneuploidy amplification confirmed by FISH in 76%. 16 of these had amplification, and 14 had aneuploidy, which correlated with strong EGFR expression by immunohistochemistry. They did not see any activating mutations in PDGFR-A or B or EGFR in the 6 cases they sequenced. In addition, they looked at coactivation of the pathways and found that 7 tumors showed strong coactivation of both PDGFR-A and EGFR. 6 of these had also phosphorylated PDGFR-B. 4 of them also had phosphorylated EPHB2, and 3 had low-level phosphorylation of AXL. They had no detectable activation of KIT, VEGFR, or the Sarc family. Among the most frequent targeted proteins with phosphorylation, they saw CREB, ENOS, TP53, JUN, STAT5B, and AMPK alpha 1 as being downstream targets. In 2014, Neuville et al. Analyzed 100 different cardiac sarcomas and tried to classify by their morphology and genetic alterations. They defined intimal sarcomas as being those sarcomas that had MGM2 amplification and found that 42% of cardiac sarcomas fit this criteria. And these could mimic a variety of other sarcoma types morphologically. 26% of their cardiac sarcomas were angiosarcomas, and 22 were considered to be undifferentiated sarcomas that did not have MGM2 amplification. They performed array CGH on 7 cases, including 2 UPS that did not have MGM2 amplification, and they found that these 2 cases had a complex genomic profile with alterations on all chromosomes, but without chromosome 12q, without chromosome 12q 13 to 14 amplification. They also looked at 3 intimal sarcomas that had MGM2 amplification and 2 that had uninterpretable FISH, and all of these had overexpression of MGM2 by immunohistochemistry, and all showed complex genomic profiles with alterations on all chromosomes, some of which were shared, specifically MGM2 amplification, PDGFRA amplification, gains of EGFR, and loss of CDKN2A, as illustrated here with this histogram showing the most common gains and losses across the tumors. Ito et al. Looked at a single case of intimal sarcoma by whole sequencing and found, again, the usual suspects: amplification of PDGFRA, amplification of MGM2, CDK4, and STAT6, amplification of CCNE1, and loss of CDKN2A. They also performed whole exome sequencing and looked at non-synonymous mutations and found that there were 63 of these, including 57 missense mutations, of which the most significant for them was PDGFRB, mutation in exon 17. Deval et al. Reported their institutional experience with 13 intimal sarcomas of blood vessels and heart using FISH. They found that the most common amplification was in PDGFRA in 11 out of 13 cases. MGM2 was slightly less common in 9 out of 11 cases. EGFR was seen in 3 out of 6 cases, and KIT was amplified in 3 out of 4 cases in which it was examined. Co-amplification was frequently seen between PDGFRA and MDM2. There was no real pattern between which tumors had co-amplification and which tumors did not have co-amplification of the different markers they examined. Fu et al. Looked at a single case of left atrium using MSK impact and found only alterations in 4 genes, including PDGFRB, which was associated with phosphorylated PDGFRB expression. They also found alterations in KMT2D, MPL, as well as an alteration in ZFHX3 that was found to be a normal variant. They saw amplification of chromosome 4q, 12q15, and 19p13, as well as the usual deletion in CDKN2. Interestingly, Zhu et al. In 2018 found metabolic defects leading to a primary PS. They performed whole exome sequencing and found 86 germline and 12 somatic variants in their patient, including a paternally derived germline mutation in fumarate hydratase. And this was a novel variant that was located in the protein binding site for tetramer formation, which led to defects in the enzyme activity. And in turn, this led to depletion of FH protein expression and increase in cellular fumarate levels. The patient also had a maternally derived germline mutation in ACOX2, which was associated with decreased beta oxidation of fatty acids. And finally, the patient had a somatic PI3 kinase mutation, which led to increased phosphorylation of AKT compared to normal tissues. And they concluded that the patient's metabolic defects, as well as the PI3 kinase mutation, contributed to the formation of this intimal sarcoma of the. In terms of copy number variation, they had loss of RGS7, MFSD1, and MARCH1, and duplication of HIF1 alpha, which correlate with protein expression. They also saw a single copy deletions of CDKN2A and PALB2, but these did not lead to differential expression of either of these genes, showing that there's alternative pathways besides PDGFRA and PDGFRB that can lead to cardiac sarcomas, indicating that in some instances metabolic activity may be. Rosick et al. In 2009, the AACR Genie database for 13 patients. We don't actually know where these arose in the cardiac wall or in the blood vessels. And again, they saw the usual amplifications in chromosome 12q. They also reported amplification of TERT, Notch2 or BB3, as well as the usual copy number losses in CDKN2A and 2B in 25%. They looked for non-synonymous mutations and found 38 of these, including 33 missense mutations. They found 2 different PDGFRB mutations in 2 patients and found that these one of the patients that had a PDGFRB mutation also had a concurrent mutation in PTCH1 and CDKN1A, as illustrated here. That is patient number 7, I believe. In terms of fusions, they found 2 fusions. One was a Notch2 PDE4DIP fusion, which was associated with Notch2 copy number gain, and also an MRPS30-ARID2 fusion. And interestingly, both these cases that had fusions were associated with PDGFRB mutations. Whether or not this is coincidence or not, I do not know. Another single case report, whole transcriptomics of a single case of cardiac sarcoma. They did not report any mutations or of interest, in quotes, so we don't know if they found anything that was not of interest for targeted therapy. And looked at expression profile of this one case versus 5 vascular intimal sarcomas and 2 angiosarcomas, and reported that it formed its own cluster, or it was distinct from both of these 2 different tumor types, but it's hard to draw conclusions from a single case. Salvador Coloma tried to look for actionable targets in 5 by a SNP array, but were only able to successfully sequence 3 of them. The first one showed a fairly quiet genome with a few copy number gains and a single loss, as illustrated here in the circus plot. Overall, very little going on. In their case 2 showed slightly more copy number gains. They found a gain in chromosome 8, a lot of very focal amplifications and losses, including chromosome 12q, MDM2 amplification, TERT amplification as well, and a few losses in various genes. And then their third case had very complex genomic profile with whole copy number gains of many chromosomes and whole copy number losses, as well as various partial losses of different chromosomes as well. You can see here the red and blue for gains and losses occurring pretty much across all chromosomes. They found specifically amplification of chromosome 8q, including PLAG1, and loss of heterozygosity in chromosomes 4, 12p, 12q, and 18q. In 2021, Kolsch et al. Looked at DNA methylation signatures in intimal sarcomas and cardiac sarcomas. They found that, as usual, MDM2 was one of the most frequently altered genes in intimal sarcomas and cardiac sarcomas, regardless of location. However, in 2 tumors that did not have MDM2 amplification, they found MDM4 amplification, and in 2 tumors they found CDK6 amplification. There were still 6 tumors that did not have any alterations in these factors, but otherwise looked very similar, with frequent losses of CDKN2B and amplification of PDGFRA. Other genes that were often altered in these tumors included cyclin D1, HDAC9, and TERT. And as they reported, alterations in MDM2, MDM4, and CDK6 were mutually exclusive, implying these probably all happens very similar. They performed a t-SNE analysis of their DNA methylation data against other histologic mimics or other tumors that had MDM2 amplification. They found that the cardiac sarcomas, the vascular intimal sarcomas, all formed a very distinct cluster all on their own, regardless of whether they had MDM2 amplification or not. And this was also distinct from other tumors with MDM2 amplification, including both low-grade osteosarcomas as well as well-differentiated liposarcomas or UPS of other sites. In 2002, Cui et al. Looked at sequencing in 9 cases. They performed FISH for MDM2, CDK4, CDKN2A, P53, and RB, and found MDM2 amplification in 6 out of 8 cases. Of the 2 cases that did not have amplification, 1 had a P53 mutation, and 1 had CDKN2A homozygous deletion. They performed targeted sequencing on 5 of their 9 cases and found 109 missense mutations, 3 stop gain alterations, 3 in-frame indels, and 4 frame shift indels among those 5 patients, with MAG recurrent alterations. However, they didn't really analyze if these were normal variants or if these were in fact pathogenic alterations, and so I'm not really certain how functional this collagen 7A1 alteration really is, for instance. They also reported on all the mutations from other series that had come before them later on. Nassif et al. Also included 18 cases of intimal sarcoma in their analysis of pansarcoma. They included 5 cardiac intimal sarcomas, 2 pulmonary artery ones, and 8 of other sites. And they found that TERT amplification was quite frequent in intimal sarcomas, including 44% of their cases. And this was interesting because TERT amplification is much more frequent in intimal sarcomas than it is in any other sarcoma type. Most other sarcomas typically have TERT activation via promoter mutation, or some cases have fusions leading to TERT overexpression. So it's a very distinct feature that characterizes sarcomas. They also looked at epigenetic pathway alterations, and intimal sarcoma here circled in red. You can see they have alterations in PRC1, MLL34, and H3K36 methylation, as well as in the SWE/SNF complex alterations. So there's some potential epigenetic alterations that are in common among intimal sarcomas. Going back to the papers looking at the most common amplifications, with a caveat that the table may be a little off because I cannot guarantee that some of these papers are not reusing the same tumors from one another, given that their MSK impact is probably included in the gene database, for instance. But overall, we can see that both cardiac undifferentiated sarcomas and vascular intimal sarcomas have MDM2 amplification in about 70 to 75% of cases. When they have MDM2 amplification, they are quite likely to have CDK4 co-amplification as well. Many tumors, about 58%, have PDGFR amplification or polysomy, again, regardless of site and regardless of whether or not MDM2 is amplified. CDKN2A/B loss is seen at about 30%, and it's again independent of MDM2 amplification. But CDKN2A loss is quite honestly common across a variety of sarcomas and is not really a defining feature of any of them. TERT amplification is quite common, as we just mentioned. HDAC amplification as well can be seen in intimal sarcomas at all sites. And some other alterations such as cyclin D1, MDM4, and CDK6. In terms of focal amplifications, we talk about 12q1 through 15. We often cite MDM2. There's others of interest that are not well studied. For example, BEST3 is involved in cardiac smooth muscle differentiation, and that's also at the edge of the amplicon and maybe also important in some cases. HMGA2, DDIT3, STAT6, GLI1, and FRS2 all have different roles and may be amplified in different intimal sarcomas and may have impacts in these individual cases. There's different mechanisms for altering TP53 in intimal sarcomas. MDM2 may be the most common one, but also MDM4 and CDK6 are seen in some tumors, as well as TP53 mutation. We found that PDGFRA and EGFR amplification do correlate with protein expression and pathway activation, so we know that receptor tyrosine kinases are quite important in the pathogenesis of these tumors. However, KIT is amplified with PDGFRA in many cases, but is not correlated with protein expression. We also have seen that TERT amplifications are common, but we don't really understand their effect on tumors. In many other tumor types, TERT overexpression is associated with poor prognosis and worse differentiation, but this has not been really studied in intimal sarcomas, particularly because they already have a poor prognosis regardless of the cell. Other common gains that have not been well discussed in intimal sarcomas include 17p, 17q, 7q, 3.1, 20q, 5p, and 8q, which have been described in various papers, but really haven't gone into depth into their impacts on tumor biology. In terms of point mutations, intimal sarcomas are copy number driven, but they do occasionally have mutations, particularly in PDGFRB, probably in tumors that do not have PDGFRB amplification. They also occasionally show TP53 mutation, as well as occasional other alterations in typical cancer genes. So overall, genomic studies have not really approached intimal sarcomas in a very comprehensive fashion. There have been lots of small series using somewhat limited techniques such as array CGH and targeted sequencing. And these studies have been never limited by the technologies. Other series are limited by their inclusion or exclusion criteria, whether or not they consider intimal sarcomas to include those that do not have MDM2 amplification, or if they can only consider intimal sarcomas to be those that arise in blood vessels. So further characterization of these tumors will probably benefit from a more systematic survey, including both undifferentiated cardiac sarcomas and vascular sarcomas, regardless of whether or not they have MDM2 amplification, and using multi-omic arrays to assess these tumors genomically and proteomically. Overall, intimal sarcomas are copy number driven. They have variably complex genomes, with gains often more common than losses, low mutational burdens, and no histologic or apparent genomic distinction based on their site, whether they arise in blood vessels or in the heart. and they have a distinctive methylation profile relative to other sarcomas, which define them as probably a single entity. I would conclude that intimal sarcoma is as good a name as any, regardless of whether they arise in the heart or in a blood vessel. It should probably be applied regardless of MDM2 amplification status for any UPS arising within the intima of the blood vessel or within the endocardium of the heart. So hopefully you've now seen what knowledge gaps we have, and we can help to work on filling this in future studies. I'm happy to take any questions when we reach the discussion period. Thank you.